In this video, we will discuss about role of mTOR in the survival and the cellular growth rate. So, in the previous video, we have understood that during the proliferation, differentiation, migrations, they have seen that genes that are related to the mTOR has gone off. Though, and mTOR is usually categorized into two parts, that is mTOR1 and mTOR2. They are two different genes. So mTOR2 is already been known to promote cellular growth rate and proliferations, but they are surprised by the genes that are related to the mTOR1 are also upregulated during the proliferations, differentiations, and migrations. They wanted to check whether this mTOR1 also has a role towards the survival and cellular growth rate of the cells or not. So first, what they did is they make the individual knockout cell lines of the genes that are related to mTOR1 and also to this mTOR2. And what they are able to find out is that when they check the cell density, they are able to see that cells in which this knockdown is occurred is showing high growth rate or high cell density compared to the control case where there is no knockdown, where there is no guide RNAs knockdown is done. No guide RNAs knockdown is done. And in the other case, when they check that after differentiations, usually the cell density goes down because of the apoptosis. However, for the control, they can see the cell density goes down, and even for this TSC1, which is related to this mTOR2, whereas when they used it for the checked it for the genes that are related to the mTOR pathway, they are able to see that their cell densities are still more compared to the control case. Thereafter, they wanted to see whether this increase in cell density is because of mTOR1 or not. So they used a reagent, that is rapamycin, at different concentration to see whether this growth rate comes down or not in the cell lines in which genes related to the mTOR1 are being knocked down. So for the control, they can see that cell growth rate remains same when they use this rapamycins at different concentrations, and when they use these rapamycins in the genes related to the mTOR1 is been knocked down, that is FLCN guide RNAs and mTOR1 guide RNAs. For FLCN guide RNAs, they can see that the decrease is there with like increased concentrations of the decrease is there with increased concentrations of these rapamycins compared to the 0 nanomolar rapamycins, both for these FLCN guide RNAs knockdown cells and mTOR1 guide RNAs knockdown cells. And so this indicates that is the mTOR1 also plays a role towards survival and cellular growth rate on proliferations, differentiations, and migrations, similar to the mTOR2, which has already been well established previously. So this is one of the new findings that researchers are able to find out that about the role of mTOR1. And in order to do that, how that things come into their mind, the mTOR1 also plays a role is from the bioinformatics analysis that they did from the proliferations, differentiations, and migrations assays, genes that are differentiated. Here, this is the main point of inceptions of the idea that mTOR1 also plays a role. And here are the genes that are differentially regulated related to the mTOR1, that is shown here, and they knock down this mTOR1 and also this FLCN, I guess, here the FLCN, and then checked its functions by inhibiting this mTOR1, also using this rapamycin, so they can see a decrease in the growth rate. So this is how they come to a point that mTOR1 also has a role towards survival and cellular growth rate along with this mTOR2. So I hope you understood these results that are presented here, and how they relate to the previous videos that you discussed. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.